Good evening, everyone. I'd like to take this opportunity to thank IIP Latur, all the respected members that are sitting, senior faculty. I'm really congratulations, heartiest congratulations to the organizing committee. We're having so many members and it's been a really successful organization. Congratulations and thank you for inviting me over to deliver this talk on gut dysbiosis. To be honest, when I got this topic, I was not really keen on it. I was like, do you want something on chronic abdominal pain? Do you want something on constipation? Those are the topics that most pediatricians would want to, and I really get a lot of patience. But then, thank you for giving me this topic, because as I went through a lot of articles and text, I really enjoyed, and what I'd be doing in this presentation is basically sharing whatever I've read. The objectives for this presentation: to know what is the gut microbiome, microbiota, the factors that influence, to know what gut dysbiosis is and its association with diseases, and this application of this knowledge to whatever extent is possible in health promotion, prevention, and treatment. This is all thanks to the genetic boom that we are having, the next generation sequencing, the 16S rRNA technology that we have got, that we have been able to detect what exactly we are having in our own intestines and our own body. Adult human body contains trillions of microbial cells. That is 10 times more than the number of human cells that we are having. So we are probably having more bacteria and more microorganisms than we have our own cells. Of these, bacteria are the most abundant colonizers, but we also have archaea, fungi, and viruses in almost all healthy individuals. What is healthy is still not very clear. So this is, like, we have starting from the stomach to the rectum and the anal canal. As we can see, the number of microorganisms goes on increasing as we go distally, and the microbiota is also varying. Like, we are starting from Streptococcus, Prevotella, Lactobacillus in the stomach, and when we go towards the colon, we have got Firmicutes, Bacteroides, Actinobacteria, Bifidobacteria. So the variety is varying, the number is varying. This is the composition that we may get in an average healthy adult, which may vary person to person and in the same person. So this is just to get an idea. Eukaryotes, human beings are eukaryotes. We have got archaea, which were initially thought to be bacteria, and you've got bacteria. In bacteria, the circle is giving, the below circle is giving an idea. These are 6 or 7 main phyla that are present in our intestines: Bacteroidetes, Firmicutes, Proteobacteria, Actinobacteria, Spirochetes, Lentispirae, and Tourniquetes. So a lot of ETs are there, and they are a part of us. Just going through a few terminologies that are there and to understand their relationship. Microbiota, microbiome, these are quite a few times used interchangeably. But biota is the living organisms, whereas microbiome is the complete gene set, because we have still not been able to cultivate the organisms. They're still not cultivable. All the organisms in our intestine, it's not possible as yet for us to get the culture media where these organisms will grow. It is like a forest in our intestines, a verdant forest with lots of variety, diversity of trees, plants, shrubs, insects, and everything going on together. So the way of knowing, we are not able to cultivate, then how do we know? The genome. Thanks to NGS, next generation sequencing, we've been able to get to know what all genetic setups we are having in our intestines. Now, if we have something sitting in our body which is not harming us, we label it as a commensal. If it is mutually beneficial, we label it as a symbiont. If it is causing a disease, we call it as a pathogen. But is it that simple? Someone who is immunocompromised, a commensal or a symbiont may become an opportunistic. Someone who has taken an antibiotic loses diversity, and that organism might become pathogenic. So a better terminology nowadays considered is pathobiont, where an organism is a potential pathogen which causes disease under a given set of circumstances when the microbiota is disturbed. So it has a potential, it may be beneficial, it may be harmful, it may be lying, sitting there, quiet, quiescent. And the two things that we are commonly using and hope that they are useful to all our patients: probiotics, live microorganisms, which when administered in adequate amounts confer a health benefit on the host, and a prebiotic, which is non-digestible food ingredient, which stimulates the growth and activity of bacteria, which are claimed to be beneficial to health. So this is like the entire theater of activity. Like microbiota are the live organisms, and their activity, it is the entire environment, the proteins, peptides, lipids, signals, chemicals, the entire environment will become the microbiome. Biome is essentially a habitat. This has come from ecologists, actually, like those who suggest study ecosystems. So what does this massive microbial community do? What is its purpose in our body? Like, since we are harboring it, obviously throughout, like since our ancestors, probably there is some purpose or a huge purpose for them to exist with us. They compete for and generate nutrients. For example, if we are having, they'll generate short-chain fatty acids, bacteria, which will give nutrition to our colonocytes. They'll shape our immune systems. It may be positively. The immune system shapes the bacteria, the microbiota, and the microbiota shapes our immune system. They can protect against and also cause acute and chronic diseases. So it's a changing, evolving relationship that we have with our microbial community. How much they influence it? They help in mucus production. So they line the intestinal mucosa, adding protection. Maturation and continuity of epithelial monolayer, separating intestinal lumen from the submucosa. Essential qualities of inflammatory responses and maintaining homeostasis. These are the conditions which will have an impact. Like if you see, we are starting with in utero. Initially, we had thought that in utero, it's like a sterile zone, germ-free zone. But because of sequencing, what we have got to know is that the gut microbiota of the mother antenatally can also be transferred to the mother. The vaginal infections, organisms can also be transferred through the placenta to the fetus. Teeth, periodontitis, those organisms can also get transferred to the fetus. So essentially the concept that the fetus is a sterile zone is, most of the times is being challenged, though it is not proved whether they are live organisms or it is just DNA genetic material. So in utero, some influence of microbiota. Then after birth, the mode of delivery. Vaginal delivery, the flora from the vagina, the fecal flora of the mother, those would be the predominant organisms that would be present, and it is mostly the lactobacillus. If it is a cesarean delivery, it is the skin microflora that would be mostly present, like Staphylococcus and Propionobacterium. So vaginal delivery, cesarean delivery, different set of organisms that we are getting. In infancy, based on the feeding, if a baby is breastfed, human milk has got a lot of oligosaccharides. Oligosaccharides are of no nutrient value to the baby, and yet they are like accounting for like 1/3 of the solid component of human breast milk, and there must be some reason why it is having. They don't get digested, they result in production of short chain fatty acids. A lot of bifidobacteria thrive in the baby's GI tract, and it's not much diverse. The microflora of an infant is not very diverse. It is restricted to bifidobacterium, lactobacillus, and veillonella. And when we introduce complementary feeding, bacteroids and clostridiols get added on. This may be in turn influenced by antibiotics. Like when we give an antibiotic, it kills irrespective. It will kill the good flora, it will kill the bad flora, and it will result in those resistant strains harboring in our intestines. So a less diverse and a resistant microbial flora would be present, which remains for maybe a month, 2 months, in adults, but in infants, it might be shown to be like lasting for a year or so. So breastfeeding, as we have seen, host genetics also impact the microflora and the environment, like your parents, your siblings, the environment in which you live, which will also contribute to the microflora that you inherit. So basically, the baby starts with a not very diverse microflora. This goes on till 3 years, and then at 3 years of age, the baby matures and has a full adult microflora. So the interactions between gut microbes and developing immune system appear to be particularly important early in life. So these are the influences we can see. Increased short the left-hand side is the good influence, right-hand side is the influence which will because of a less diverse microflora, which is not very good and resulting in diseases. So any alteration in the intestinal microbiota can be called gut dysbiosis, but to say what is altered, we need to know what is normal, and what is normal is yet to be defined. So how do we get dysbiosis can cause disease? Possibly by alteration in the intestinal permeability, alteration of the local and systemic inflammation, and alteration of the intestinal maturation by gut microbes and their metabolites. So various acute and chronic diseases are associated with intestinal dysbiosis: necrotizing enterocolitis in preterm, late onset neonatal sepsis, antibiotic associated diarrhea, and in the chronic disorders, obesity, type 1 and type 2 diabetes, dyslipidemia, inflammatory bowel disease. How can we attempt to attenuate neonatal intestinal dysbiosis? Rational use of antibiotics, some recommendations for prebiotics and probiotics, and something which is experimental, swabbing of newborns who are born through cesarean sections with vaginal flora of the mother. These are some of the recommendations given on use of probiotics by ESPGHAN in 2022. They are available online. So very few positive recommendations are available for certain conditions like acute gastroenteritis. Here the point made by the committee is that probiotics should be strain specific, not just the species. They should be strain specific, should be given in the dose in which they were studied. The durations are not yet defined. These are the recommendations given for necrotizing enterocolitis. Huge 15-point recommendations are made for use of probiotics and NEC in 2020. So wishing a happy and healthy gut to all of you. Thank you so much.